hello everybody. How you doing out there in SPF world? I'm SPF Tim, and I'm glad you're all with us today. Hey, I wanted to say thank you to everybody for participating in the HSP and PLS Awareness Week. We had a lot of time, as you can see. I have check marked all of the days of the week that we've done so far. We're down here at the bottom. We made it to Saturday. We made it through the week. It's been a great week, meeting a lot of new people, having a lot of fun, talking to a lot of people, spreading the word and awareness about HSP and PLS to all of our friends and families and people that we meet, getting the word out there so people can enjoy helping us on our journeys. I wanted to welcome everyone to the Spastic Paraplegia Foundation's 2021 virtual annual conference presentation today. This event is the fourth of 6 in a series scheduled over the summer and fall, highlighting the researchers and doctors that work to find a cure for HSP and PLS. We are using the regular Zoom platform today, but we can control your microphones and cameras. We are going to leave your cameras on, and later in the show, we're going to let everyone go live so we can have some time to visit together.
I'll put your salad in the refrigerator.
Hey, thank you very much for doing that, salad person. I love that. If you need to move around, please turn your camera off, okay? Like Mary Schultz is doing up there in the background. Hey, our guest speaker today will be answering some of your questions after his presentation. So you must use the chat box on the bottom of your Zoom screen to send in your questions. Also, we are collecting your questions today. So if we don't get to all of them, our guests will try to answer them at a later date. Hey, also, we're going to cut off the questions at 3 o'clock today, okay? That way we can have this in a nice tight box. Also, please note that we're recording today's virtual conference presentation, and it will be available on the SPF YouTube channel soon. Also, we want you guys to stay with us after the presentation because you're going to get some more information on future events, and we've got the 5K awards that are going to be handed out today, okay? So we need you to stay with us after the presentation, all the questions and answers. Stay with us because there's a lot of good stuff that's going to follow up after that. So remember, we're going to leave your cameras on today. We're going to control your microphones, okay? And then we'll let everybody on a little bit later in the show, okay? So everybody sit back, relax, have a great time today. It's now my honor to introduce the president of the Spastic Paraplegia Foundation, Mr. Frank Davis.
Well, hello, everyone. It is my sincere pleasure today to introduce to you guys today an old friend and SPF board colleague, Dr. Corey Braastad. Corey has been on our board for over 15 years and is the only person ever on our board without either HSP or PLS. The reason he is such a vital part of our board of directors is that he is an expert in genetics, and we all know that genetics science is becoming an ever more important part of finding a cure for HSP, and it has become even more important for PLS as well. Corey is not only a learned expert, but a tremendous human being. Corey Braastad completed his PhD at the very distinguished Brown University in the Department of Molecular and Cellular Biology and Biochemistry. He completed his postdoctoral training at the University of Massachusetts Medical School. He then moved into industry at Athena Diagnostics, where he developed diagnostic genetic tests for rare diseases. That is how Corey connected with the Spastic Paraplegia Foundation, and he was invited to join our board over 15 years ago. And time again, he has been one of our most popular annual conference speakers. 5 years ago, Corey joined LabCorp Drug Development to lead genomics testing for global clinical trials. Please join me in welcoming Dr. Corey Braastad.
All right, it's a pleasure. Thank you for inviting me to join today. I usually get feedback from each conference that people want to hear about some of the things that I've spoken about in previous years. And usually for a scientist, that's kind of like death, right? You don't want to keep talking about the same thing. But for me, I really enjoy talking about complex topics to have a broader audience understand. And we all need to understand about our own conditions and about our own disease, because with rare disease, you have to be your own expert, right? There are very few experts in the world. So my only job, as far as I'm concerned, these past 15 years, is to make sure that everyone on this call, and usually in person, but on this call, is equipped to be able to either ask the right questions, to get in contact with people that can help get you answers to your questions. And in that way, today, one of the most important parts of my talk will be the question and answer sessions. And it sounds like we're going to be able to submit questions for us to talk about in the chat. And Tina, I think that you're going to be kind enough to go through, in case I miss early through our presentation. If I miss something in the chat, Tina's going to help me make sure that I don't miss it. So we can be sure to answer important questions. And at the very end, of course, we can always summarize and follow up offline if we need to. I've got 3 parts of my talk today, and I'll get to that in a moment, but I just wanted to talk a little bit personally about kind of, how much it means to me that, I'm part of your community. And more recently, this past year has been a bit of an eventful year for the entire world, right? It's been such a challenge. There's been a couple of extra challenges, I think, with our community. And in some ways, I've joined the rare disease community in kind of a new way. I did have a diagnosis just about this time last year of early onset Parkinson's. And so I'm kind of working through that diagnosis this year personally. So I'm now part of the neurodegenerative disease community and working through in a different way than you all, but certainly it's something that I'm appreciating in a new way now. And I'm feeling great, right? A big part of this is exercise. And so I'm trying to, learn how to box and beef up my muscles. So let me see if some of you may have seen that. So I wanted to make sure that you know that I'm addressing this head on and happy to answer questions about that too. Let me see if I can share my screen. Let's see, share. And let me know. Does that come up? Is that a full screen view of my slides or only a partial? Does it look okay? Does it look okay?
We see your notes, sir.
Okay, let me switch that display setting. I'm sorry. I may need to try that again. And how is that?
We see all of the threads on the left-hand side, all of the screen. So you need to wide screen it. There you go. Yes, sir. Perfect.
Does that work better? Okay. All right. Sorry about that. All right. I think that we're ready. And if you see anything different, let me know. So I'll have 3 parts of my talk today, but they're going to be somewhat brief. I've discussed basic genetics in the past, but we always get feedback that people do want to continue to hear and get a primer on basic genetics. If you, like I, dialed into the first couple of virtual sessions, you heard some pretty complex terminology. And I think that the speakers were extraordinary at talking to an audience of patients like us. But there's always some terms that can go over your head. And I want to make sure that we address that as much as I can. So I will give, even though we have past presentations that you can see on our YouTube channel, I do want to spend a little bit of time talking about some basic genetics terms, a little bit of the lingo, and make sure that if there's questions, we get those answered. The second part of my talk will be a cell and gene therapy update. And I've done this the past few years too, but obviously this is an area that's always changing. this is actually what I do for my work as well, right? I run clinical trials for cell and gene therapy. I'm involved deeply with some of these projects that I'll talk about. Some I cannot talk about, but I will be able to give an overview and obviously answer any questions. I want to especially make it relevant for our disease areas, but it's really important that you see what's happening in the world in this area of cell and gene therapy. I will finish up by giving a review of the virtual conferences that have happened so far this year. I think it's useful to kind of look backwards and give a bit of a snapshot of what I saw. I had done this, I think, in San Antonio in one of the recent meetings, and that was received pretty well, right? If I can kind of summarize, I haven't been able to see Dr. Fink's talk, unfortunately, because I was out that day, but I'll be able to review Dr. Paganoni's and Dr. Oslinder's talk. And moving forward, I can also give some updates when we publish. So those are the three parts of my talks: basic genetics, the cell and gene therapy update, and then a bit of a review of some of the virtual conferences that we've heard so far. So starting with basic genetics, I love to do this in person, and you all probably know that because I like to invite the audience to come up and stand up and be part of, DNA and a protein code. And not being able to do that, I'm still going to review some of the terminology today. So as you may or may not know, DNA is the genetic blueprint, and it's up there in the title, right? That is a pretty fundamental piece of information, right? That is what DNA is. DNA stands for deoxyribonucleic acid, just a fancy way of saying DNA. You don't have to worry about that long term. But any abbreviation in this talk, I'm going to try to give what the expanded view is, because otherwise it can be a little bit difficult to interpret. So DNA is the genetic blueprint. All of our DNA is located in the nucleus of every single cell that's in our body. And again, in our bodies we have, and I think you can see my pointer. Let me see if I can pointer options. I might need to make a laser pointer. Can you see that laser pointer?
Yes, sir.
And okay, fantastic, thanks. And with my my Parkinson's, I might have a little bit of shakiness there, but that's something that we're all used to, and that's okay. If it was live, I'll try not to blind anybody next time I see you all in the audience. But we're all safe looking at it on Zoom. So if you look at this as a diagram of a cell, within the cell, there are actually structures. And you've probably seen this if you saw, for instance, Dr. Oslinder's talk. She was talking quite a lot about these structures that are inside of cells. And I'll be able to talk more about those as well. She was talking about mitochondria, which are the powerhouse of the cell. She was talking about endoplasmic reticulum, which helps make proteins. Well, the nucleus is also a part of the cell, and that's where all of your DNA is, right? So all of this DNA that's located in every cell is in those nuclei. And for these cells, you can see this is kind of like a blobby structure, but every cell looks a little different. You've got about 3 trillion cells in your body, absolutely huge number. And these cells are actually turning over probably about every 3 months. Some turn over a lot slower. Some, like actually the retina of your eye, never turn over. Those are with you for the rest of your life. That's why it's important to protect that. I'm sorry, the lens of your eye, not the retina, but the lens of your eye. Very difficult to repair. But most cells are actually dividing and repairing themselves. But these cells look very different based upon where they are in their body. So for instance, a hair follicle, right? It doesn't look globular like this. It actually has a structure that actually supports growing a hair out of it. A liver cell looks very different from a bone marrow cell. And that looks very different from a nerve cell, right? Like you've seen these structures in the past couple of talks of, for instance, upper motor neurons, where they have very long spindly structures. These cells can look very different based upon where they are in the body. But the things that are common about them is, for instance, inside the nuclei of each of these cells is DNA, so that cells can make a copy of themselves. So DNA is actually existing wrapped up in structures called chromosomes. And you've probably heard of the term chromosomes. You may not know what it is, but this is approximately what a chromosome looks like. It kind of looks like an X. And if you take this chromosome and you stretch it way out, like is depicted here, if you take it and stretch it, it's very compacted, because there's a whole lot of DNA that needs to be in a small amount of space. If you stretch it out, it ends up having a lot of different structure, right? So chromosome is in a structure, and you've got this histones in a structure, and then you've got this, what you probably traditionally have heard of DNA is in a double helix. And I actually got a tattoo of a double helix on my arm. That's how much of a DNA geek I am. That was my first tattoo this year. So you got this DNA in a double helix. What you have in each one of these cells are 46 of these chromosomes that are in 23 pairs. 23 come from mom, 23 come from dad, and you end up with a complete complement of 46 chromosomes in each cell. It's a huge amount of material that's fit into something that's smaller than the dot of a pencil. If you look at the structure of DNA, again, you take that double helix and you actually untwist it. So here's your twisted helix. And if you take a look at it, kind of flattened out, it can't exist like that in nature. In nature, the lowest energy state is for it to be twisted and in a very nice structure that's very stable. But if you take it and twist it out, you end up seeing that the building blocks of DNA are called nucleotides. And you've probably heard A's, G's, T's, and C's. What this is a code, okay? So each of these nucleotides looks and feels a little bit different, and they actually stick together. So A's stick with T's, G's stick with C's, and so you've got two halves of DNA that actually stick together really, really tightly, almost like if you've had two pieces of glass with water in between, and how it's really hard to pull them apart. That's very similar to how DNA is on a molecular level, right? There's lots of bonding between these A's and T's, G's and C's, and it makes, again, a very tight structure, and that spirals on itself. These A's, G's, T's, and C's are actually the code that makes DNA a blueprint. It's what directs what happens next, and I'll go on to the next slide for that. So if you look at the DNA code, right here, again, a chromosome that gets wound and it's spiraled into a code, A's, G's, T's, and C's. You can actually see that this is indeed a code that you can read like a book. It's very much like a book. In fact, I'd almost call it, it's almost an outdated reference, but I'll use it. It's like a telephone book, because if you're reading, you can actually see that there are sections of the telephone book that are called genes, that are like a section where your house might be, for instance, or almost like an address on a street, okay? And the AGTC, all it is a code. And this code gets read and transcribed into RNA. And I don't want to get too complex into the words transcription and translation, but I don't want to ignore the term RNA, because like DNA, RNA is a temporary version of this code, and that's right here. So A's, G's, T's, and C's become U's, A's, C's, and G's. And you've heard probably of RNA recently, because two of the major vaccines that were made against SARS-CoV-2 were made through mRNA technology. And so I've kind of glossed over this in some of our recent conferences, but I don't want to miss the fact that mRNA is a natural part of reading this code. And you've probably read about it in the paper and maybe wondered about it, so I didn't want to gloss over it today. So if there's questions, let me know. But it's this RNA that is read in groups of 3, and these 3s are called codons, and that is how you get translation into amino acids, all right? So you've got a DNA code that goes into an RNA code that then gets made into an amino acid code. And that's how DNA drives the manufacture of proteins. Let me go to the next slide to get a little bit more about this. All right. So a gene is a unit of hereditary information, and it occupies a fixed position on a chromosome. So we saw those chromosomes, those 46 chromosomes, 23 that come from mom, 23 that come from dad. And at each of those addresses, kind of like your home address on your street, you'll have a gene. And I didn't want to ignore the word gene here, because as we get into things like cell and gene therapy, you hear a lot about genes. There are certain genes that have function that can actually cause disease. And what's important for you to know over here is that genes code for proteins, and it's those proteins that have structure and function. And when I talk about structure and function in person, I sometimes refer to things like, you know, the building of a house, like the two, sorry about my dog, there must be a delivery here. Proteins have structure and function, kind of like 2x4s in a house, allow you to have a wall that has structure on it that you can hang things on, that you can build on, it supports the roof. Things like a light switch, right, that has a function, you actually flip it. Some proteins actually are able to have shapes like a key and lock, for instance, that actually do things in your cells to make things happen. What's important to know is that when the DNA sequence changes, the protein sequence changes, and that can actually alter the protein structure. So as you go from DNA to RNA, and then eventually into this amino acid chain into proteins, those proteins fold up and form a function. And if something changes in the DNA, we actually figured out back in the 50s what the impact is going to be on the protein, how that code changes. And if something changes, it could cause disease. Now, in a lot of cases, it does not cause disease because there's plenty of variation. We all look a little bit different, and that's because our DNA is all a little bit different, but there can be changes that impact proteins that cause them to not function correctly or to gain a new function that's incorrect, and that can cause disease, especially in rare disease. And this is a little bit more. This is where I would actually stand people up if we were together in a room. I would stand people up to become the chains of a protein, right? And we would actually talk through. But if you look again at these DNA codons that get translated into an amino acid chain, you've probably heard of amino acids because they're in things like, you know, shampoo and stuff like that. And amino acids are in, you know, vitamins. You know, these are things that we ingest and you're probably familiar with some of the, you're probably familiar with tryptophan because you hear about it during Thanksgiving, that's an ingredient in turkey muscles that can make you a little bit sleepy. So tryptophan is an amino acid. And if you look at the sequence of amino acids, in this case, this is a serine, valine, lysine, cysteine, tyrosine, glycine. They all are a little bit different in their structure. And when they're together in a chain, they fold into a protein that can be like a 2x4 or like a microtubule in the case of the SPG4, or like I said, kind of like you can have almost like a lock and key shape to something. And if something changes about one of those amino acids, it's the thing that can cause disease, okay? So proteins have structure and function. When the DNA sequence changes, protein structure and function can be altered. And I don't mean to go too fast, but I do have almost entire hour-long presentations on this. So if you have more questions, feel free to bring those to the Q&A, or we can refer back to some of the YouTube channel videos from previous years. All right, I'll have one more slide on other types of changes that can happen in DNA. So back here, I'm looking at individual changes within a gene, a particular gene. But you can have other types of changes in DNA. So remember back to our chromosome structure? Some of them are in the form of an X, but this is just a simplified version where it's just the X's together in one I form. You can have a deletion where an entire section is missing. And do you think that that might be impactful in terms of association with disease? Yeah, it definitely can. Maybe not, depends on how big it is, but a deleted area could have an impact on disease. You can have a translocation where you actually have a region that is supposed to be in this direction and it's flipped instead, right? Or you can have an entire arm that goes onto the tail of another chromosome. And that might be in fact impactful as well. It might not be in some cases, but most of the time it will be impactful. And the inversion is where you can actually have a region flip, and that could be impactful. And then, of course, there's those single gene changes, much like what we were just talking about, where maybe one single nucleotide, one of those letters of DNA changes that can cause a protein change. And that could be a small segment that can cause an issue or cause disease. And that's what you order when you order a molecular test for your disease. We'll be looking at those types of specifics. And if there's questions, please make sure that you note that in the chat. Unfortunately, I am not looking at the chat right now, so I'll answer all the questions at the end, but I think the team is looking for your questions, and I'll get to those toward the end. So I don't want to spend too much time on that, but I don't want to skip over what is usually some pretty difficult terminology. So let me take a few minutes now just to give you a little bit of an update on cell and gene therapy. I think you've seen me in the past almost get a little emotional about this kind of slide, because although in some ways this slide is not big enough to satisfy all of our needs for therapy today, I would say that this is a list that is longer than I expected a few years ago. So I think it was back in 2017 I started making this slide. We had these 4 approved drugs that have just recently been approved that pretty much changed the patient community for spinal muscular atrophy and for cure of a genetic, very rare form of blindness, and 2 types of cancers. That was the tip of the iceberg at that point. And we do now have 4 additional approved therapies, 3 of them literally from within the past few months, that are in the same category of cell and gene therapy. It's remarkable. It's good progress. It's not fast enough, but it is an incredible tide that has shifted. So I think you can see here under cell therapies, these are all different kinds of therapies. And on my, I think 2 slides from now, I'll go through the definition of each one of these. And if you have additional questions, be sure to mention them at the chat at the end. But I'll talk about antisense oligos. I'll talk about what in the world is CAR T for fighting, mostly that's for fighting cancer right now, but there are definitely other implications. I'll go through what CAR T looks like, what's happening in developing that kind of therapy, what's taken out of your body, what's modified, how that fights cancer. I'll go through that in a slide later. And there's an example, of course, of 2 different types of gene replacement, one to cure that blindness that I mentioned, and a gene replacement for spinal muscular atrophy, which is an upper motor neuron disease, and the impacts that that's had on this patient community. Some of these can get quite pricey, but there are advocacy groups, much like ours, that have been very important for helping make sure that these are covered by insurance for all appropriate scenarios. And that's in many ways would be the next evolution of a group like ours. First, we have to drive for therapy. Then we have to make sure that there are things like affordability for all of us. So there's plenty of work left to do, but it's good progress. So let me take a few moments to go through, this is from the Alliance of Regenerative Medicine, a really important partner for me in my job. I work with a lot of these clients, to be perfectly honest with you. So if you look at the cell and gene therapy state of the industry, you've got about 1,200, more than 1,200 cell and gene therapy clinical trials going on globally today. And I work on a number of these, and this is a slide that was actually put together by my company because we are making sure that all of our experts within my company are driven and focused on doing everything we can to move this work forward. As I just showed you, there's 8 FDA-approved cell and gene therapies. Those are all within the last 4 years. There was nothing before that. And of course, 3 of those are within 2021. If I've shown some of the money in the past couple of years, but this is a bit eye-popping. And I think this really, if you think of putting your money where your mouth is, 19.9 billion of global financing has been in the area of cell and gene therapy. This is not some afterthought of the pharmaceutical industry. This is front and center, and this is an enormous investment in cell and gene therapy. To me, this shows that there is commitment to moving major programs forward. We see that certainly with this 1,220 clinical trials, but this is an eye-popping number, and I certainly expect this to grow. 9.9 or 9.8 billion raised just in 2019, and so far this year we're keeping pace to increase that. I'll talk a little bit about phases of trials. I've done that in the past, so I don't want to spend too much time on it, but I don't want to talk about trial phases without giving some context. But a phase 2 trial means this is happening in patients, okay? This is impacting patients. It does not mean it's a cure. A clinical trial is an experiment in people, right? And we know a little bit more about that since we've heard some of the terms about vaccines and emergency approval and things this year. It's been a little unusual of a year for that. So maybe people do understand that a phase 2 trial means that it's in people. It means that it's past minimum safety criteria, and this is treated in a small group of actual patients to look to see if there's response. And there's 685 of those in various diseases. That is tremendous and did not exist just a few years ago. So this is huge promise for what's to come. Doesn't mean that the 685 trials will be successful. It doesn't mean that all of those drugs or even most of those drugs will be approved, but it does mean that there's an enormous tidal wave of work occurring now that is going to impact a lot of people in a positive way. Late-stage clinical trials continue to advance. But the expectation, again, by this Alliance for Regenerative Medicine, this is a really important group, and it's actually a group that I think the SPF is going to need to get closer with. What they expect is between 10 to 20 new FDA approvals per year by 2025. And 2025 is not that far away. It's right around the corner. We've got 8 so far. You've kind of seen how this grows, right? We're going to be advancing at a tremendous clip within a matter of a few years. These are not in the areas of all of our diseases that are impacted by us on this call, but this means that more and more of our disease patient community will be impacted, and it becomes a matter of time. And it means that there's an entire machine within the biopharmaceutical companies that are going to be more and more capable of delivering effective therapy soon. We have to make sure that we get our place in that pipeline and in that R&D pipeline, but this means that there's FDA commitment. Let me go through, again, I said that I would define what each of those different classes of therapies are. So let me go through a little bit of that cell and gene therapy lingo. Some of this you may have seen already, and so I do not mean to keep going over this each year, but I do think that it's important that we all be on the same page. So some types of cell and gene therapies. Here's a few, and you've probably seen some of this in the news. Antisense oligos, ASO. This was the first treatment in spinal muscular atrophy. And as it turns out, and some of what I mentioned last year about N-Lorem, they're the group that originally did ASOs, antisense oligos, in SMA. And they may have, there's applications that they're exploring, especially with Dr. Fink and others, to have applications in some of the HSP genes, which can be really, really exciting, even for, really personalized medicine where, it's not a big patient community, but just a few people that have a particular change. So antisense oligos are worth keeping in mind. I'll go through CAR T, a slide of this later. So CAR T stands for chimeric antigen receptor T cell therapy. This is especially useful in cancer today, and I'll show it. You take out cells from your bone marrow and modify, put them back in, and they go after your cancer in a very targeted way. It's only appropriate for certain types of cancer, but it's incredibly effective if you have that type of cancer. And I've got a few examples of that later. And this can be expanded beyond cancers, but for right now, most of the breakthroughs have been in fighting oncology and cancer. Another word that I'm sure most of you know is CRISPR. A lot of people have questions about this, but think of this as gene editing. Remember back to when I talked about genes? Genes are at a particular hereditary unit at a particular place on a chromosome, like your house on the address of a street, right? And if you've got a gene that's making the wrong protein, this is a way to, in some cases, edit that gene to restore it to normal function, to make the correct protein again. And that sounds easy, but it's taken a long time, longer than I expected, to have that be effective and safe in humans. And we have, I'll show a little bit later on, where we have effectively treated, I think at this point, over 22 people, for instance, that have sickle cell anemia and beta thalassemia, some pretty common, well, common in some areas, blood disorders that can be very impactful. And with CRISPR editing, we've had our first very successful patients that have all responded and are at this point effectively cured of their disease by using CRISPR. Very exciting, and that could be the future for a lot of people on this call. It'll take time, but it could be very effective. So unlike editing a single part of a gene, if you think of gene replacement therapy, you're taking, for instance, your whole house, right? The whole gene, and actually replacing it with a fully functional form. So instead of making an edit of just one base pair, one nucleotide, an A, G, T, or C, or maybe a couple, you're replacing the entire sequence so that it makes this kind of block. It's a different way to get that genetic material into the appropriate cells in your body, either by making an edit in the existing genome or by delivering a genetic payload into those cells. And an example of that kind of gene replacement therapy is we've actually got somebody on the board, Karina on the board, who started a Maddie Foundation for her daughter with SPG15. We've actually had some grants awarded from the SPF, and they've been instrumental in trying to get a gene replacement therapy program going for SPG15. And there are plenty of other people, and I think it's a very tight community now within the SPF community that has SPG15 that may benefit and that are really paying very close attention there. So it's exciting. And it's exciting to have all of these different possibilities happening in parallel. We don't want to put all of our eggs into any one basket. That's something that as a scientist, I think is very important. And we can talk about that too. You may have heard of pluripotent stem cells. Stem cell research is not advancing at a rapid pace, and I don't at this point think that there's a lot of viable options. Certainly none that I could clinically, I would say, are at any kind of clinical stage. I think that there's some hype around this, and I worry about some people even being tempted by possible snake oil, where you may be recruited to alternative countries, or claims of miraculous types of treatments that I don't think are necessarily clinically proven at this point. There can be debate about that. I'm fine with that, but there isn't much clinical evidence here. There's definitely important uses for stem cells, but there isn't too much of an update there in terms of direct applications into therapy at this point. I added mRNA therapies at the end. This, as I mentioned earlier, has been made a lot more popular with the successes of both the Moderna and Pfizer SARS-CoV-2 vaccines. But the therapeutic implications of this kind of mRNA technology is a lot broader than just vaccines, and a heck of a lot broader than just infectious disease. And I think that this is going to be a really important area that's going to get it's gotten a real shot in the arm, no pun intended, well, maybe pun intended, to expand this kind of impact into other therapeutic areas than just vaccines. All right, let me go through a little bit about what is cell and gene therapy and what does it do. And I've shown a similar slide before, so I'm going to go through this pretty quickly, but basically altering the DNA sequence of an organism for purposes of for treating disease is what's called gene therapy. And because it's happening in cells, some of this nomenclature is changing a bit. It's not just called gene therapy anymore. It's called cell and gene therapy, because the unit of action is really a cell. You want cells to be healthy. If you've heard Dr. Odslingser's talk, it's about cells, right? Keeping cells happy. And certainly that was a message also from Dr. Paganoni, right? When she's seeing inflammation of neurons, it's about the cell as a group, right? Genes are what drive proteins to be expressed in cells, but it's the cells that you really want happy and fully functional. That's going to prevent disease. An important challenge of cell and gene therapy is targeting the correct areas. So as you can imagine, it's very difficult to target, for instance, in your brain, the upper motor neuron cells. So be able to target those areas is important. One of the reasons there's been successes, for instance, with gene therapy in the eye is because it's accessible. You can bathe the eye in, the correct sort of therapies, and it's accessible, right? You can get it. It's like if you have skin cancer and it's easier to treat, it's the same sort of idea. Some areas like lung are very difficult to get at, and there's not a lot of nerves. It's hard to understand. So, to be able to get at that is really important, but we're getting better and better at that kind of targeting. Some of the therapeutic genetic material can be delivered to cells via something called an adenoviral vector, or AAV. You may even see this in the paper now. So I don't want to get too technical about this, but to talk about things like AAV is important. That's for delivering things like gene replacement therapy, where you've got a big genetic payload, right? This is like the FedEx truck that is delivering a big package to a cell. But if you've got a little package, like maybe this is more like a letter carrier instead of a big package that's heavy, like a little letter carrier, you can have a lipid nanoparticle for very small genetic payloads like antisense oligos that are extremely small and not bulky in size. And I've talked about CRISPR previously, so I'll leave that on the screen, but this is a natural biological process, and you're making small edits. And again, you need something like this AAV, adenoviral vector, to deliver something as large as CRISPR to be able to actually make an edit, but you're basically making some very small changes within a gene. These are just a couple of case studies. Again, these were put together by my company because we have a very close connection with many different diseases. And these are 4 people with whom we are quite familiar. So Humberto was treated with for non-Hodgkin's lymphoma with a therapy called Yescarta. This was after his normal chemo course had failed, and he is thriving, and it's amazing to see. Hannah's a little girl who is treated for that hereditary blindness that I talked about before. She was 2 weeks old at the time. She was 2 weeks old when she was diagnosed. She was treated with Luxturna at age 7. I believe a few years ago, actually showed a video of someone, I don't think that it was her, but it was someone similar to her, who after treatment could actually navigate a maze as part of their therapy and could do it, where before treatment, they could not get past the first step of the maze. It's an amazing change. She's now running around playing outside with her brothers and sisters. Matteo, I actually, I'm on the board of directors for spinal muscular atrophy called Cure SMA, and Zolgensma, the gene replacement therapy, was delivered to Matteo at 27 days old, and he's now got aspirations to be an astronaut. It's changed that community dramatically. Mary was diagnosed with multiple myeloma, treated, this is a therapy that was approved just a matter of months ago. After previous treatments have failed, she had the right kind of multiple myeloma to respond to this new drug, and she has had an incredible response. Just a few examples of what we can enjoy in the future. This is a bit more of an expanded list, but I would say it's a partial list because remember, there's 1,220 of these studies going on literally today. I don't know about all of them, but I do know about a lot of them, and I'm involved with many. You see our 8 approvals up top so far. And then I picked just a few, some of which I've mentioned, like gene editing with CRISPR in sickle cell and beta thalassemia. That's in phase 2 trials right now. The companies, there's actually several companies that are doing this work, Vertex and Bluebird among them. And I can't talk too much about my involvement, but I'm deeply involved. This is not approved, so we don't have a cost here, but for gene replacement therapies, you're usually talking about almost a couple $2 million. That's been the price tag. But again, it's important that you understand that insurance companies are picking up the tabs for these in a very real way. And that's important work that our patient advocacy group will be doing on behalf of our patients when it gets to this time, working with the FDA, working with payers, working with standards of care, working with physicians. These are networks that we are looking to set up, and we do this together. This is not an individual problem. This is us coming together as a community. There's also tremendous work moving forward in other antisense oligos, again, from companies like Ionis, who did that work with SMA and ALS, which of course is a very close cousin to PLS, with a couple of different genes, SOD1, HTT for Huntington's. Amazing work going on there. Another gene replacement therapy for the same gene, SOD1 for ALS, and from Avexis and Novartis. And again, I mentioned earlier work toward SPG 15 that's sponsored in part by the Maddie Foundation and the Spastic Paraplegia Foundation, of course. So these are just the tip of the iceberg, and there's a lot more that's happening than just this brief list, but these are some that are relevant for us. I mentioned a little bit about phases of FDA-approved clinical trials, and I just wanted to touch on that. But I've mentioned this before, so I don't want to dwell on this. But I did want to have it in the slide deck so that at least when we see, you can pause on this slide and understand a little bit more and be able to answer questions. So if you remember, any sort of testing in humans is called a clinical trial, right? It is not a cure. It is a trial. It is an experiment. And it's all about safety. So a phased trial is 100% about trying to make sure that it's safe for people, but that we're not moving so slowly that we're creating more harm. So if you talk about a phase 1, the endpoint is all about safety. So usually this is a test after it's been done usually in animal models, sometimes in non-human primates, to make sure that this is safe to actually be able to put into a person. And it's a test to see if there are issues that can arise. So you're really looking at safety profiles, making sure that you're doing basic testing to make sure that things are not going badly. It can be for all sorts of different indications. You can have single dose, multiple dose, escalation of doses to make sure that dosing is reasonable. As you get into a phase 2 trial, you're really looking at efficacy and starting to see in patients, is there an appropriate improvement? And that's where you need to know. And if you listen to the previous talks, you heard about biomarkers and you heard about, some sort of indication that there's a benefit, right? That isn't just a self-reported benefit, that you're actually getting an outcome measure that can sit that any scientist or the FDA can look at and say, oh, look, this is where they would be without treatment. Here's where they are with treatment. There's an improvement here. You got to be able to measure something. If it's self-reported, sometimes we can fool ourselves and you can have a placebo effect. So often there is a placebo arm. So you can have 2 arms where there's no treatment or a placebo treatment and then a treatment of the drug. You can compare between the two as well. So that can happen in a phase 2. And then with phase 3, usually this becomes a much larger cohort. But of course, in a very rare disease population like ours, we might not ever be able to do a phase 3. We might really have to rethink what these phases are and have it be a phase 2. And I think that the FDA is coming very far in agreeing with that sort of approach, and that especially for rare diseases where it's a small patient group, you're not going to be able to get a 20,000 person study. It's not possible and it's not feasible. So phase 3 maybe for other diseases like, diabetes and flu and things like that. Right? Phase 4 is usually monitoring post-approval. So once, these drugs that we showed previously that were approved, if there are reported issues of anything that pops up. I mean, you hear about that, for instance, with, the vaccine in the news, right? There are patient reports of issues, and if those add up, we can put a halt to a particular drug trial because you don't want to cause additional harm that maybe as we treat more people, we find something that can happen that maybe we hadn't found before. So phase 4 is all about monitoring and making sure that there's good efficacy. And I'll move on. If there's questions, I can address that, but I'll move on from there. I wanted to go through a little bit because this is a new slide that you haven't seen, a little bit about CAR T and T cell therapy. So that's where there's been quite a few approvals of this, especially for the treatment of cancer. And I wanted to be able to talk a little bit more about this. So the way that CAR T works, and remember, this is chimeric antigen therapy. So you actually are able to get cells by removing T cells from a person. This is just from intravenous blood, right? So you're removing a patient's own blood, and you're then taking T cells, which is a subset of your immune cells, and you're inserting a gene to make what's called a new chimeric antigen receptor. And what that does is it depends upon the kind of cancer that you're going after. That allows that T cell, that immune cell, to seek and destroy cancer. So you make that modification. You're making it specifically to go after a personalized kind of cancer. You're then growing that by the millions in the lab. This is part of the drug manufacturing. This is personalized medicine to a T, right? You're taking those cells out, you're making them so that they can seek and destroy, and then you grow up millions of those CAR T cells and you put them back into the person. And those CAR T cells go find that cancer and they destroy it. And it's personalized and it's incredibly effective, but only to people that have this particular kind of cancer. Does that make sense? Okay. And if there's questions about that, let me know. Now, obviously, this can be important for areas outside of oncology, but this is most impactful right now to oncology because a lot of these patients would otherwise die. And so there's been a lot of FDA movement forward to be able to do trials in folks that otherwise don't have options, and that's resulted in approvals of those drugs. But the benefit for everyone else in the field and all of us is that we've been able to learn a lot about how to make CAR T cells, what kind of dosing, how many millions, what are the safety issues? Do these CAR T, you're putting in actual live cells that are changed. Is that going to cause some problem 20 years down the road? We know a lot more about that now, and we're continuing to monitor these patients that have responded and would otherwise have likely have died by now. But we can monitor and understand how that impacts them longer term, and that can those are learnings that are important for the community, so that we can apply this to other areas of disease. Okay, that's my update in cell and gene therapy. And let me just go back to that previous slide. I don't want to go back, but I want to go back a little bit. This is important. So 1,220 cell and gene therapy clinical trials are going on right now globally, and 685 of those are in phase 2. These are happening in patients with these diseases today. That's really important. Targeting 10 to 20 FDA approvals per year by 2025. Okay, and if there's questions, I'm looking forward to seeing those in the chat. All right, for the last part of my talk, and I'll spend probably about 15 minutes on this, and then we can open up for questions, and maybe even a little bit less, and then want to leave plenty of time for questions. I wanted to share with you a few of my notes that I got from our virtual conference talks so far. I think we've had an extraordinary lineup. We usually have an extraordinary lineup. This has been a particularly good lineup. I think very highly of all of our speakers so far. First, I just want to talk a little bit, we've heard from Frank, our SPF leadership, in each of the sessions so far. And, some of the major take homes that I'm hearing from him, and I'd love to hear more from him, is that, we're a group of 135,000 people working together for Cures in Our Lifetime. It's amazing to me, and I see that Jim Sheehan is going to be talking a little bit more about some of our fundraising efforts later and giving some awards for virtual 5Ks. I don't think any of us would have thought that, a few years ago, that our 2021 goal is to break 1 million in donations for the first time ever. That's amazing, and it's a real tribute to all of you, and it's a tribute to a lot of the great folks that we have working on behalf of the SPF today, and it's amazing. Frank noted that this burden of $1 million is actually coming from only about 11 to 12% of us, and imagine how much more we could accomplish if more of us donated. If it was 30% or 50%, or like you said, even 95%. There's a lot that we can do, and all of this research is being driven by dollars. Words that have echoed in me from Frank is that we are a force for good, and that we are actually a force for incredible good. Absolutely true, and I love that sentiment. I listened to Dr. Paganoni with great interest. She is an amazing person. For those of you that don't know, I think many of you do. Of course, we all know Dr. Paganoni is brilliant in the area of PLS, and she works in the area of ALS as well. But one of the struggles we've always had as the SPF is that we raise a lot of money from both the PLS side and the HSP side, but the PLS side raises a ton of money. And there aren't a lot of well-known genes, for instance, that drive PLS. The research is further behind because it's more rare, it's been more challenging. And one of the ways that the SPF wanted to bolster research in this area is not just by, usually we award grants, right, that are based upon really good feedback from the scientific advisory board, really rated very high that it's going to be impactful. One of the things that we did a number of years ago, I think it was maybe even 8 years ago now, Dr. Paganoni was one of the recipients, is we made a fellowship in collaboration with the Niels Foundation that she now is an important part of the leadership of, to foster and help sponsor a new trainee that would be focusing on PLS, with the idea that they probably would keep focusing on ALS later in their career. And holy mackerel, has that paid off in spades for Dr. Paganoni. I mean, she's passionate. She's a leader in the field. She's amazing. And I don't want to say it's because of the SPF, because she's brilliant in it, but we did have the foresight to be able to invest in that area and try to foster that kind of work. And that's really worked extremely well. And there's a lot of people in the SPF leadership that are to thank for that, especially the people that donated the money for that. So let me go to a little bit of a review of what Dr. Paganoni touched on. Go see her talk. It's on the YouTube channel. It's a little bit less than an hour. It's phenomenal. If you haven't seen it, it's phenomenal. Her title is What's New in PLS? And my takeaway, this is my note, is it turns out a lot is new. She talked about, in very easy to understand terms, about a very steady and impressive increase in published research papers on PLS. She actually showed a timeline, of how many papers are published each year. And it's so impressive to see. It is like 411 total papers, and a lot of them are from the last few years. The trajectory is going skyrocketing. She talked about the fact that the immediate goal for PLS is really to be ready for clinical trials, right? We just had an update on cell and gene therapy trials, right? It's exploding. We want PLS to be in that mix. We want HSP to be in that mix, but this is focused on PLS. She was saying that to be ready for more trials, significance been made by researchers who've been partly supported by, you know, the SPF for many years. And the SPF has really enabled this research in 3 major areas. So Dr. Mitsumoto from Columbia, we've supported for years, right? This is a major colleague of ours and of Dr. Paganoni's. She talked about the big impact from Dr. Mitsumoto to lead an effort to redefine PLS diagnostic criteria in a very helpful and practical way. This has been really important to understand, you know, if you have PLS, if you don't have PLS, it's important to distinguish those. She talked about probable PLS as an important part of that diagnostic criteria. So that's really important to understand a correct diagnosis. Second, she talked about prospective registries and defining a natural history. She said this was also led by Dr. Mitsumoto. So it's not work just done by him, it's with a group, but it's important leadership by him. And an area where Dr. Paganoni is pretty humble, but I know she's made huge strides, is in new tools and biomarkers that have been developed. Some of those biomarkers that she talked about allow PLS to be a lot more trial ready when possible treatments are available. And she specifically went into imaging of neuroinflammation. And some of these pictures are really cool to see. And that promise, I think she probably is just quite humble about it. I think that's a very important biomarker for PLS. And some of this was not known until very recently. So she's really, she and some of her colleagues at Mass General Hospital have been right on the forefront of making this happen. The inflammation patterns are very different between ALS and PLS. She describes that in her talk, and certainly very different with HSP, where there really isn't much neuroinflammation. I also heard from Dr. Paganoni in her talk that, you know, she's extending an invitation to PLS patients to connect with her and discuss getting some of that imaging at Mass General Hospital. And there were other parts of her talk that are notable, but those are my major takeaways. And I would encourage everyone that wants to see it to go take a look at that talk. I've got a couple of slides here from Dr. Hande Odswinner. She had a multi-part talk with a lot of information. She is incredibly active in multiple areas of upper motor neuron treatment and biomarkers. And so let me give you a few of my takeaways from what we heard from Dr. Odswinner. So her title was Building Effective Treatment Strategies for Upper Motor Neuron Disease. There was a couple of Q&A breaks. So her outline, you know, she went through what is an upper motor neuron and why do they matter. And just like I talked a little bit about cells earlier and how they're all a little bit different, she gave a really good overview of what is special about an upper motor neuron and what makes it difficult and what are some of the amazing opportunities and just more about it. So if you want to learn more about upper motor neurons, definitely refer to that section. She then talked about what are the roadblocks for having an effective treatment strategy for upper motor neuron diseases. She then went on to talk about overcoming some of those barriers for effective treatment strategies. Then she went on and finished with, you know, what's the path for developing cures for upper motor neuron diseases, and she has some really interesting approaches there. So starting up at the top, right? So she talked a lot about how mouse neurons are a really good model for upper motor neuron disease. And she made a distinction between mice and people versus upper motor neurons in mice and people. So, you know, something that's effective in a mouse is not necessarily effective in a human, but she drew this analogy that really mouse upper motor neurons are very similar and behave very similarly between mice and humans. And so when you're specifically looking at upper motor neurons, it's a very effective model. So she's done a lot of work in these kinds of mice that are defective for the various genes that we know well that cause our disease and she's looking at those in a mouse model. She's taking an omics approach. And what I mean by that is, remember I was talking about DNA, and I was talking about RNA, and I was talking about proteins. If you talk, we kind of joke to ourselves in the genomics research community that we call it omics because we're talking about genomics and proteomics and metabolomics, and it just kind of gets almost comical that we call everything omics. So, but that's what you're looking at. You're looking at large sets of data where you're looking at all of the DNA, where you're looking at all of the RNA, or you're looking at, you know, all of the methylation in a particular cell or in a particular area, or, you know, some area of focus. So she talks a lot about doing omics analysis very broadly in a lot of these kinds of disease tissues. And then she also talks about doing electron microscopy to actually, instead of just looking at some of the pieces of it, right, the proteins and so forth, but to actually look and visualize the structures to see what might be wrong or what might be right. And saw that there's quite a few abnormal findings, and a lot of others have as well, right? I mean, we've heard from Dr. Craig Blackstone, just brilliant mind in this area that recently went from the NIH into Mass General. But you know, he's looking at a lot of visualization of these structures too, and she's finding that there's abnormal structural findings in various mouse upper motor neurons. A lot in mitochondria, as you would expect, where there's, you know, a lot of energy production in different kinds of vesicles that are important for protein transport, especially when your neuron is so long, it can literally be huge. You've got to be able to transport material around. So she saw abnormalities in some of those vesicles, and certainly a lot of abnormalities in endoplasmic reticulum, which is important for protein production. She then went on to talk about quite a bit of work with protein-protein interactions in disease models. She then switched gears to talk, instead of looking inside cells, she talked about, you know, instead of intrinsic factors, she talked about extrinsic factors, things that, you know, are around upper motor neurons, like things like astrocytes that she was talking about that can impact upper motor neurons. So I think she's really taken a holistic approach, inside, outside, and then beyond. She also made an important point. There won't necessarily be a single drug that will cure all upper motor neuron diseases, because there may be a few different mechanisms of action. And she really takes, she takes a lot of time to distinguish between, you know, a mechanism-based approach for therapy as opposed to a disease-specific approach. And I think that's definitely smart and worth looking at. Sorry, I duplicated a few of those at the top of this slide. So she went into a really interesting analysis of a cell-based platform that she and her lab had developed to find drugs that target specific mechanisms of action with a healthy cellular structure as an outcome measure. So she's actually looking in disease tissues in upper motor neurons in mice to say, all right, what makes them better, right? What makes healthy structures? And she gave as an example, it's made a big media splash, especially with our community, this NU9 as an example of a potential therapeutic. But I also think that she would be quick to point out that you don't necessarily want to put all of your eggs in one basket. She's trying to look broader, and she tested a lot of different kinds of compounds to be as broad as possible about what can be a potential therapeutic. I'm sure that she's still looking for others. It's not like, oh, we found it, we're done, and then go on. She's still looking for sure, right? But NU9 is interesting to carry through and see, all right, it looks like this might make structures, cellular structures in mice look more healthy. Might that be the case in humans? So as she looks at NU-9 treatment of upper motor neuron in diseased mice, it seems to restore healthy cellular structures, including mitochondria and endoplasmic reticulum, which, as we said, is important for energy production, important for protein production. Some of the next steps are toxicology studies to make sure that this is safe, right? But before we risk putting any of these kinds of compounds into people, we have to make sure that they're safe. And an FDA application to start that process is important as next steps. That's the sort of thing that takes money. It's the sort of thing that, you know, places like the SPF can help support, and other places too. And then, you know, there's definitely plans for phase 1 trials to determine safety profile. And we don't know how far off that is. I think that she actually got a few really good questions about that, about timeline. And I think, you know, that's the sort of thing we'll be waiting and seeing to see how fast we can go. And I can't wait to get an update from her at some point soon. She also finished up with some non-human primate, I think it was a macaque model for viral delivery directly to upper motor neurons. So that's an area of work for her as well. Remember, I said that targeting of the particular kind of cells is really important to get the therapy into the right spot. She was doing some work there. Unfortunately, I had to miss Dr. John Fink's talk. We don't yet have that posted on the YouTube channel, so I will post my review of that soon. John usually does an amazing, I mean, everyone in these conferences always rate him top. I mean, obviously he is our medical leader and always has been, and he is a phenomenal resource in the field. I'm not a clinician. He is the clinician, right? A clinician of all clinicians. So, you know, obviously he always gives an amazing state of the art, and especially asks very personal or answers very personal questions of members of the audience. So I expect that to be amazing, and I look forward to seeing that soon. And that is all that I have, and I want to take on the most important section, which is to answer any and all questions that might be out there amongst us all. And with that, I'll stop sharing. So I am going up. So it was probably about 20 minutes ago. I see a question: if somebody has chemotherapy, what gets killed, especially if the cancer comes back? Okay, so I can talk a little bit about that. So in terms of chemotherapy, and again, I am not a clinician. So this is the lab geek side of me and things that I've learned, but I am not an oncologist, but I do know some about the mechanisms of how chemotherapeutic agents target cancerous cells. So I don't want to go too far down this rabbit hole, but I will. So the question is, if a person has chemotherapy, what gets killed, especially if the cancer comes back? And so if you think of cancer, it is our own cells reverting to a state from when you're growing as a baby. So when you're growing as a baby, what I would call in utero, right, in mom's belly, your cells are growing and growing and growing and growing and growing as fast as you possibly can. Later, as your organs mature, there are brakes put on cell division because you don't want too much cell division. Cells need to repair, they always need to be growing, but you want to put a halt on it, all right? But when you're growing as a baby, you need to be growing a lot. So a lot of cancer that comes is those brakes get taken off. So if you think of like a car, you put on the brakes, and if something happens with your brakes and you're speeding down that hill, think of that as cancer. And the body is very good at targeting cancers and getting rid of them, especially with your immune system. We've learned quite a bit about that. When you have chemotherapy, you're generally trying to kill cells that are dividing fast. So back, 30 years ago, when you're given things like radiation, that is to stop any cell in your body that's growing fast. And you happen to kill cancer at the same time, but that's why it would kill like a lot of your stomach lining, and you would have nausea. That's why it would, it would kill more cells than just those that you're trying to target. So you would kill quite a few. But as we talk about CAR T cells, it's not going after rapidly dividing cells. It's going after the cancers, those things that have hallmarks about those cancers that are unique, things that are expressed on the cell surface, things that are unique. But that's why it's personalized medicine. And these therapies are going after those cancers like a laser, and they're destroying it and not other stuff, so it can have less side effects. But there can be additional side effects because it's stimulating your immune system. You can have things called like cytokine storms, where it almost feels like a flu. So a lot of people that have these kind of CAR T therapies, they have an immune response, and it can feel like you're sick, but you're not. It's not because you're having lots of cell death all over the place. It's because you're having a specific immune response. And there are ways that we can control that, and that becomes an important part. And if there's a question about that, a follow-up question, let me know. Next question from Tim, it looks like, is how do we get into some of these phase 2 trials? And it's really important that you talk to your neurologist about willingness to be in phase 2 trials, and make sure that you have a neurologist that has the right connections to get into these kinds of trials. If they're knowledgeable about trials, and there's centers of excellence like in Boston, like in Seattle, like in, you see down in Baylor, and I mean, there's places all over the country that are just amazing with clinical trials. You really have to have neurologists that are engaged with those kinds of centers, and most neurologists are. The problem is that there aren't many phase 2 trials in our specific disease groups now, but be ready, talk to your neurologist next time, you have an appointment with them. When I talk to my neurologist, I've got a top-notch movement disorder specialist at Mass General. I've talked to her about enrolling in trials. Absolutely. And that's going to be your connection. Next question was at 3:01 p.m. and I'm just reading through the chat here. And if there are, please, other people can come off of mute and we can have a discourse and we can have video, that's fine, but I'll just keep going through these questions. Hello, recently a new friend of ours, Julie from New York, has SPG5A. Oh, interesting. Okay. So SPG5A is one of those interesting cases. I probably want to have you talk to Dr. Fink. Okay, so she has a question. Could SPG5A complex cause optic neuropathy? She's been legally blind for 6 years now. Second part of the question is the medication Hannah used an option for Julie? Really good question. Okay, so optic neuropathy. Definitely talk to Dr. Fink about this. I'm not the clinician here. I know about 5A, but he knows more about the medical implications of it. I know more about what's going on in cells and how some of these folks can actually respond to bile acids effectively, but he would be much more, or your neurologist, of course, would know more about the clinical pieces. However, could it cause optic neuropathy? You need to talk to him about that. Could the medication that Hannah used be an option? I would say emphatically, not likely. And I say that because it's specific for a particular gene mutation. And the gene, I think that it's RET9A, something like that. It's a specific gene, not SPG5A. It is not specific for that gene. And so the thing with gene therapy is, if I, for instance, had that gene therapy, it would likely be, I would be unaffected because it's correcting something that isn't defective in me, and that would correct something that isn't defective in Julie, for instance. Okay, so just because it's effective in Hannah does not mean to be effective in Julie, and that's why they call this cell and gene therapy personalized medicine. In some cases, it is extremely personal, and what helps one person may not help another. But it's worth looking into more of the details. But I would say that that's not likely.
Dr. Corey, I think
please, Tina.
Okay, I think you spoke really well about Dr. Ostendler's NU9, but you can wrap that into the next one from Hank. How
Sure.
does NU9 affect specific known SPG genes or ALS known genes? I understand it may help upper motor neurons. How are they related?
One of the really exciting things about her research, and specifically how they found NU9, is that they were using the health of a cell as the outcome measure, not looking at a molecular pathway. I'm extremely interested to know what the molecular mechanism of action is for NU9. And I mean, she may know, and maybe she's, still doing work before she publishes on it, but I'm guessing that they're still trying to figure out what the actual effect of NU9 is molecularly. They know that it's causing healthier structures in mouse upper motor neurons, but I don't think they know the molecular mechanism. So I don't think it's exactly clear yet if that's going to help with HSPs. It looks like it may, because it's all for upper motor neuron health, but I don't think that it's clear yet which specific diseases would be helped. I think we're all hoping that it would help them all, but until we know more information, I don't think that we know. So as a scientist, I've got to be pretty comfortable saying I'm not sure yet. And in this case, I'm not sure yet, and I can't wait to know more.
Well,
And that's where I
Dr.
think I
Corey,
would put myself as kind of, cautiously optimistic, right?
Dr.
Sorry, go
Corey,
ahead.
that's the last question that is in the chat. You were really good with giving a concise summary of all of the presentations before. I thank you so much.
My pleasure.
I just wanted to share some information with you about our virtual 5K. This is the third year that we have participated with this. So far, as earlier this morning, we have 38 people that have raised over $28,000. That's a pretty good jump from last year's amount. So there's 3 participants that have just started to participate. So hopefully we will be able to reach our goal of $35,000. It's not too late to start. If you have second thoughts, would like to get involved and help us raise the money, please let me know. I can be glad to share with you the easy steps. The fundraising page is really quick and easy to do. So the top 5 fundraisers so far: Jackie Wellman has raised $5,675. Christopher Falconer has raised $3,245. Dina Lampairs raised $2,537. Tammy Monahan has raised $2,045. Emily Smeltz has raised $1,955. So that's lots of money. So thank you guys for doing that. Now the top 3 fundraisers will be getting a medallion similar to this, something like this in the mail. And then also those that raise at least $100 or more will be getting a shirt like the one I'm wearing, a light blue shirt with the logo. So if you're participating and you've not raised your $100 yet, please do so as soon as possible. I'm going to give the awards out based on money raised by next Saturday, September 4th. And also I'll go ahead and order the t-shirts for those that have raised $100 or more by then. So if you have any questions or problems, please let me know. Top 2 teams are the Crogans, Tina and Tim. They've raised $2,350, and John and Jody Cobb have raised $1,325. So that's our 2 biggest teams that have participated. So even though we're going to kind of wind down the fundraising as far as the awards and shirts by next Saturday, fundraising can keep going on. That you can keep going through the end of the year if you like. So if you want to participate, please get on, sign up, and go forward and do what you can. We've had a lot of unique participants this year. You may have seen Mary Schultz rode her bike over 150 miles. I mean, that's just amazing to do something like that. Gina Fraser swam. She swam her 5K, which is pretty cool. And we had several people that used adaptive devices to do their 5K. So lots of these pictures and posts are on Facebook. So if you have social media, check those out. And with time, we will be sharing this information with you online on our website and so forth. So look for those in the future. I'm sure with the COVID surge and stuff like that, that's probably had a negative impact on some folks raising money. So if now you didn't raise as much money as you thought or couldn't raise any money at all, we are sorry for that. And if now's not a good time, then maybe you can find a way to help us out by the end of the year. So please keep that in mind. So overall, we have raised $450,000. That's a large chunk of change. That is almost enough to fund 3 medical research grants. So that's a chunk of change that we can do that. So if you've contributed already, thank you so much. If you've not, hopefully you can find a way to put us in your budget by year's end so that we can hopefully raise $1 million for the first time in SPF history. That would just be amazing.